Antigen antibody reactions. Babatha Elias, antigens and antibodies combine with each other specifically and in an observable manner. In the body, they form the basis of antibody-mediated immunity in infectious diseases, or hypersensitivity and autoimmune diseases. In laboratory, they help in diagnosis of infections, in epidemiological surveys, in the identification of infectious agents, enzymes. Antigen antibody reactions in vitro are known as serological reactions. Stages of AG/AB reactions. Primary stage. Initial interaction between AG and AB, invisible. Rapid, occurs at low temperatures and obeys the general laws of physical chemistry and thermodynamics. Reaction is reversible. AG and AB is bound to each other by weak van der Waals forces, ionic bonds and hydrogen bonding. Secondary stage. Demonstrable events, precipitation, agglutination, lysis of cells, killing of live antigens, neutralization of toxins, complement fixation, immobilization of motile organisms and enhancement of phagocytosis. Precipitin, AB participate in precipitation. Agglutinin, AB participate in agglutination. Precipitinogen, AG participate in precipitation. Agglutinogen, AG participate in agglutination. Tertiary stage. Includes neutralization or destruction of injurious agents or tissue damage. Also includes humoral immunity against infectious diseases as well as clinical allergy and other immunological diseases. General features of AG/AB reactions. The reaction is specific. Entire molecules react and not the fragments. There is no denaturation of the antigen or antibody during the reaction. The combination occurs at the surface, so surface antigens are immunologically relevant. The combination is firm but reversible. The firmness is influenced by the affinity and avidity of the reaction. Affinity refers to the intensity of attraction between the antigen and antibody molecules. It is the function of closeness of fit between the epitope and antigen binding region of its AB. Avidity, strength of the bond after the formation AG/AB complexes. 6. Both AGs and ABs participate in the formation of agglutinates or precipitates. 7. Antigens and antibodies can combine in varying proportions. Both AGs and ABs are multivalent. Measurement of antigen and antibody. Measurement may be in terms of mass or more commonly as units or titer. The antibody titer of a serum is the highest dilution of the serum which shows an observable reactions with the antigen in a particular test. Two important parameters in serological tests, sensitivity and specificity. Sensitivity, the ability of the test to detect even very minute quantities of antigen or antibody. When the test is highly sensitive, false negative results may be absent or minimal. Specificity, the ability of the test to detect reactions between homologous AGs and ABs only, and with no other. In highly specific test, false positive reactions are absent or minimal. Types of antigen-antibody reactions. Precipitation reaction. Agglutination reaction. Neutralization reaction. Complement fixation test. Immobilization test. Opsonization. Immunofluorescence. Radioimmunoassay. Enzyme immunoassay, precipitation reaction. Principle: when a soluble AG combines with its AB in the presence of electrolytes, NaCl, at a suitable temperature and pH, the AG/AB complex forms an insoluble precipitate. When instead of sedimenting, the precipitate remains suspended as floccules, flocculation reaction. Precipitation can take place in liquid media or in gels such as agar, agarose or polyacrylamide. Zone phenomenon. The amount of precipitate formed is greatly influenced by the relative proportions of AGs and ABs. If increasing quantities of Ags are added to the same amount of antiserum in different tubes, precipitation is found to occur most rapidly and abundantly in the middle tubes. Preceding tubes, Ab excess, prozone. Middle tubes, Ag and Ab in equivalent proportions, zone of equivalence. Later tubes, Ag excess, postzone. Mechanism of precipitation. Merrick, 1934, proposed the lattice hypothesis, mechanism of precipitation. The multivalent antigens combine with bivalent Abs in varying proportions, depending on the Ag-Ab ratio on the reacting mixture. Precipitation results when a large lattice is formed consisting of alternating Ag and Ab. Merrick's hypothesis. Merrick's hypothesis. Applications of precipitation reaction. It can be carried out as either a quantitative or qualitative test. Sensitive for the detection of Ags. Types of precipitation reactions. Ring test. Slide test. Tube test. Immunodiffusion. Electroimmunodiffusion. Ring test. Consists of layering Ag solution over a column of antisera in a narrow tube. E.g., Ascalis thermoprecipitant test, grouping of streptococci by Lansfield technique. Slide test. When a drop of Ag and antiserum is placed on a slide and mixed by shaking, floccules will appear. E.g., VDRL test and RPR test for syphilis. Tube test. This is employed for the standardization of toxins and toxoids. Serial dilutions of toxin/toxoid are added to the tubes containing a fixed quantity of antitoxin. The amount of toxin that flocculates optimally with 1 unit of the antitoxin, LF dose. E.g., contest for syphilis, immunodiffusion, precipitation in gel. Advantages of immunodiffusion. Reaction is visible as a distinct band of precipitation. Stable, can be stained for preservation. Indicates identity, cross-reactions, non-identity between different Ags. Single diffusion in one dimension, Udine procedure. Ab is incorporated in agar gel in a test tube and Ag solution is layered over it. Ag diffuses downward through the agar gel, forming a line of precipitation. 2. Double diffusion in one dimension, Oakley-Fullthorpe procedure. Ab is incorporated in agar gel, above which is placed a column of plain agar. The Ag is layered over it. The Ag and Ab move towards each other through the intervening column of plain agar and form the precipitate. 3. Single diffusion in two dimensions, radial immunodiffusion. Here the antisera is incorporated in a gel and poured on a flat surface. Wells are cut on the surface to which Ag is added. It diffuses radially from the well and forms ring-shaped bands of precipitation concentrically around the well. 4. Double diffusion in two dimensions, Auchterlini procedure. Helps to compare different antisera and antigens directly. Agar gel is poured on a slide and wells are cut. Antiserum, central well. Different Ags in the surrounding wells. 26. Reaction of identity, lack of relatedness, partial identity. Fig 2. Lack of relatedness. Partial identity. 27. Helix gel precipitation. 28. 5. Immunoelectrophoresis. Graeber and Williams devised this technique. This involves the electrophoretic separation of composite Ag into its constituent proteins, followed by immunodiffusion against its antiserum, separate precipitin lines. It is performed on an agarose gel with an ag well and ab trough cut on it. The test serum is placed in the antigen well and electrophoresed for about 1 hour. Ab against human serum is placed in the trough and diffusion allowed for 18 to 24 hours. Immunoelectrophoresis. 31. Electroimmunodiffusion. The development of precipitin lines can be speeded up by electrically driving the ag and ab. 2 types. Counterimmunoelectrophoresis, one-dimensional double electroimmunodiffusion. Rocket electrophoresis, one-dimensional single electroimmunodiffusion. Counterimmunoelectrophoresis, CIE. This involves simultaneous electrophoresis of ag and ab in gel in opposite directions resulting in precipitation at a point between them. Produce precipitation lines within 30 minutes. Clinical application, detecting ags like alpha-fetoprotein in serum, ags of Cryptococcus and Meningococcus in the CSF. 2. Rocket electrophoresis. Used for quantitative estimation of ags. The anti-serum to the ag to be quantitated is incorporated in agarose gel on a slide. Ag in increasing concentrations, is placed in wells punched in the set gel. The ag is electrophoresed into the ab containing agarose. The pattern of immunoprecipitation resembles a rocket. Rocket electrophoresis. 35. Laurel's two-dimensional electrophoresis. Variant of rocket electrophoresis. The ag mixture is electrophoretically separated in a direction perpendicular to that of the final rocket stage. Used to quantitate each of the several ags in a mixture.